Okay, we'll go ahead and get started. And I first want to say thank you so much for joining us today. For those of you that don't know me, my name is Tresa Warner. I am number one, Lexi's mother. She has OTC. She is 27 and she was transplanted at 21. So that's my first role. And my second role is the executive director of NUCDF. I'll have to say, I have been rehearsing what I'm about to say, and I can't get through it without some tears. So,
okay,
being a mom just kind of leaks out every once in a while. So, those of you that know me, that's just, I guess, part of my charm, right? So I'll go ahead and get started. I want to thank everyone for the commitment to participate today. For those of you that have joined a family conference in the past, it's intense, but a transformational experience. We want to welcome our new families that are joining us from around the world. We have international families that are in our audience today. As we gather virtually, we designed a day with engagement in mind. You know that connections are the heart of NUCDF. And while we can't embrace in person, we're going to strive to connect in the most meaningful ways possible. And this is where the tears will come. So here's a little background to inspire us as we begin. NUCDF was founded nearly 40 years ago by families that did not have hope, and despite their despair and trying to hold on to some hope with no treatment and heartbreakingly low survival rates, they were not deterred. With unwavering determination, these families championed for the development of butanol and participated in a small clinical trial that ultimately was FDA approved. And because of that, we have treatment today. Ignited by hope, we persevered over the last 3 decades with remarkable milestones: newborn screening, an additional treatment, then another, and then another. And in the recent months, I've witnessed, and I know you have too, the wave of hope that is embodied by a little cutie pie named KJ. I know I'm not alone in expressing the surge of encouragement when we saw news reports, articles, and social media feeds just explode. I know that you probably received the messages that I got. Hey, is this story about that baby? Is that the same as Lexi? And those were the texts I had dreamed about. What KJ's journey has done to our community is it's proved that change is possible, and it's happening all around us. KJ represents just one remarkable advancement, paving the way for a brighter future for UCDs. We have active clinical trials for innovative treatments, including enzyme and mRNA therapy, along with gene editing, and many more treatments in the pipeline. Now, this has not come without frustration and profound loss. Our families know despair, yet we embrace intense joy with every triumph. From the moment we receive the diagnosis, we discover a caring network of families that welcome us, tuck us right underneath their sleeve, and walk along this journey with us. Our sense of connection is at the heart and soul of NUCDF. Many have made extraordinary contributions as pioneers in patient care and research, paving the way for us. Now, we will lead the way for others, making breakthroughs happen, just as we already have begun to do. And thank y'all for letting me whimper right through that. I'm sorry. I want to start by a little bit of housekeeping before we get started. The meeting today will be recorded. The presentations will be recorded, the discussions and the breakout sessions will not. We would like for this to be a safe space for our families to discuss in an open forum. We do have industry partners that are joining us today, and they will not join the breakout sessions. Just as like in our family conferences in person, the breakout sessions are really just for us to connect. I want to thank those that are going to help facilitate those breakout sessions. My goal is to have it like we're sitting by the pool or around a table at dinner, just discussing. We're going to use Slido. I'm a career teacher, so I believe in engagement. We're going to use some technology called Slido. as we're going through our presentations today to help with engagement, check for understanding, and see if we have any questions along the way. When you see the Slido come up, and it'll come up on the next slide, you'll be able to take your phone and scan the QR code and answer questions. Or if you have your computer, you'll be able to go to slido.com and there'll be a code that you can put in. We thought this was just a fun way to get us engaged and connected. All of the presentations, resources, links, anything that we discussed today will be available following the meeting. We want you to sit back and enjoy and not be concerned that you may have missed something. We'll make sure that everything is provided. Joining me today is our Deputy Director, Jill Williams, and our Executive Assistant, Amanda Swank. If you're having any tech issues or if you have any questions, Amanda will be monitoring the chat. When we begin the discussion section, if you know how to use, or you can use the hand raise feature on Zoom, you could use that, or you can put a question in the chat. We'd like to thank our conference sponsors today is Amgen and Ultragenyx, and they're joining, I see them in our list, they're joining us today. And we're grateful for their support, continued support of our community and this conference. We have an esteemed panel of medical professionals with us, and I want to thank them for sharing their talents with our community year after year after year. I'm going to go ahead and start with our first Slido. So if you've done this before, you'll know. If not, we'll wait a few minutes and let everybody kind of engage. You can scan the QR code, or you can go to slido.com and you can put in that code. Okay, so we're going to go ahead and get started. We will have more questions like this as we move through. This is our conference schedule. Everyone received this in an email. If you did not, we have it up on the screen for you. And then, of course, we'll make this available following the meeting. This is our esteemed expert presenters. And we do have a packed day. The goal was to try to finish as close to 4 o'clock as possible. Within each section, we will have a presentation, an opportunity for discussion, a breakout, and then a small break, and then we'll get right back into it. We do have a few breaks along the way. But we'll go ahead and do one more. And let's see, which UCD does your family represent? And you should, if you used your phone, you'll automatically see this question populate. And if you were using your computer, you'll see it on Slido. It will automatically populate. Okay, thank you everyone for participating in that poll. And we're going to go ahead and get started. Dr. Nagamani, I'll do your introduction, and then I will send the slide share over to you. Dr. Nagamani is a crowd favorite of NUCDF. If you've attended one of our conferences in the past 20 years, you know Dr. Nagamani. He is the professor and vice chair of clinical research in the Department of Molecular and Human Genetics and professor of medicine at Baylor College of Medicine in Houston, Texas. As an internist and clinical geneticist, he provides care for adults with genetic disorders. His research program is focused on clinical translational research in genetic disorders. He is the 2011 NUCDF Fellowship Awardee and is currently a medical advisory board member for NUCDF. He serves as the co-principal investigator for the NIH Rare Disease Clinical Research Network, the Urea Cycle Disorders Consortium. Dr. Nagamani works with NUCDF on research and educational initiatives and has been a constant participant at NUCDF conferences for many years. And I will turn over to Dr. Nagamani.
Again, many thanks to the NUCDF for this opportunity. And this is one of my favorite talks to give. And this is one of my most stressful talks to give, because if you bomb in front of your home audience, there's nowhere to hide and nowhere to go. So I hope I don't have to go into hiding after this talk. I am Sandesh Nagamani. I grew up within the UCD community. I have a research career focused on UCDs because of my engagement with NUCDF. And I'm honored to sort of present an overview of how life is being transformed for people with UCDs. And my elevator pitch today is to take you through from where we were to where we want to go. And I'm going to represent the work of many people. And I hopefully have acknowledged everybody along the way appropriately, but please be informed that I'm just a cheerleader for all of the work that I'm presenting here. So these are my disclosures of interest. I am fortunate to have NIH funding, again, all public taxpayer dollars of y'all's, that allows us to do our work. And I have active research collaborations with industry partners, but I have no financial conflicts to disclose. So what I'm going to be doing today is to break my talk into three parts. I'm going to give you a brief historical perspective, and that would be where we were part of the talk. I'm going to talk about the urea cycle, the urea cycle disorders and their discovery, and some data on early treatment and outcomes. I'm going to pivot to the second part to from where we were, how did our journey take us to where we are now? I'm going to talk about collaborative research network that includes all of y'all, the NUCDF, the researchers and physicians, how we went from where we were to where we are now. And then I'm going to try to present a very brief roadmap of this collaborative, where we want to go, how are we going to look at the partner network moving forward, how are we going to establish clinical care and research care networks. I've given this talk a few times, so for some of you who remember these slides, I apologize, I'm using the same ones. Again, all living things need nitrogen. Nitrogen is a component of proteins, of all tissues, of nucleic acids, and many important biological substances. Now, just like water everywhere, not a drop to drink, it's nitrogen everywhere, but not an atom to take. There's 70% of the entire atmosphere is not nitrogen, but not one atom of it enters through our breathing. So all the nitrogen that we get into our body has to come either from the food, the water, or the animal-related fluids such as milk and others. So once the nitrogen enters our body, we make amino acids and then proteins with it. We make nucleic acids with it. And so the nitrogen is used up. Now, whatever excess nitrogen is there, the one that we don't use up, collects as ammonia. Now, ammonia is not that soluble in sort of water. Now, that's not a problem if you have water all around you, right? So if you are an aquatic animal, the ammonia can just be diffused out directly. So you're excreting excess nitrogen just as ammonia per se. But when we came onto land, that is, aquatic animals came onto land, that became a water problem. You're not surrounded by water, so you need to convert ammonia into a compound that is more soluble in water. So if you're a terrestrial arthropod, lizard, or birds, you convert it into uric acid. But if you are a mammal, all humans, some adult amphibians, and some types of fish, you convert it to urea. So how is it that you take a gaseous substance of urea and convert it of ammonia and convert it into urea? So that is where the urea cycle is important. This remarkable gentleman who is on the slide, his name is Hans Krebs. He discovered the urea cycle. Even before he started working on this, it was known that the amino acid arginine can be split by bacterial enzymes to create urea and ornithine. Now, Hans Krebs were working on liver slices. He would make thin slices of liver and understand how metabolism works. So when he put extra amount of ornithine, he ended up getting increased amount of urea. That's not something you would expect. You put arginine, you get more urea, you can sort of understand. But you put more ornithine, he also got urea. Furthermore, he figured out that if you put ammonia and carbon dioxide, again, you get increased urea. So what he also noted, he was an amazing biochemist, and with the techniques that he had at that time, he still figured out that no matter how much ornithine you put or how much ammonia and carbon dioxide you put on the liver slice, while the concentration of urea increased, the concentration of ornithine never increased. So he was like, you know what? Maybe the ornithine never gets out. So it has to be in a cycle where it goes round, and urea is one of the products that exits out. So working over the next few years, he figured out the ornithine gets converted to citrulline, and then citrulline to arginine. He did not know what was in between at that point. And he was the one who discovered this cycle, which is the first cyclical pathway, metabolic pathway discovered, to say that, well, this is how we convert toxic ammonia into a relatively non-toxic product, urea. Now we know that ammonia, that is a waste product of excess nitrogen, is taken through a series of steps by these enzymes that are listed here in blue, and they're converted. The nitrogen from ammonia is trapped into these compounds, which are then converted to the next compound. Finally, we will have urea, which is relatively a safe product and can be excreted. What happens if there's a block in this process? If you don't have an enzyme or you can't get these products through the cycle, then you have a urea cycle disorder where there's excessive buildup of ammonia. And I don't need to tell any of y'all what ammonia does. You have lived experience of this, and this is your worst enemy and nightmare, as it is ours as well. So if you have a block in the OTC enzyme, then you have ammonia buildup upstream, you have OTC deficiency. If you have a block in the ASS1 enzyme, citrulline also builds up along with ammonia upstream. That's why it's called citrullinemia, because of citrulline building up. If you have a block in ASL, you have block, not only you can have the increased ammonia, but you can also have this argininosuccinic acid buildup. That's why it's also called argininosuccinic aciduria, and so on. I'm not going to go over all of the enzymes. So it took almost 20 years from the time that urea cycle was discovered to the first disorder, the urea cycle disorder, that was discovered in the 1958 in Lancet. This is a bulky compound that is not usually present. So that was the first abnormal metabolite in urine. That's why it was the first urea cycle disorder that was detected. Now, within a few years, the most common one, the OTC deficiency, was discovered. And within the next ensuing years, in the 60s, as well as in the 70s and early 80s, all the enzymes that are required for urea synthesis and their deficiencies were discovered as the causes for urea cycle disorder. Right now, these are the urea cycle disorders that we know. There are enzyme deficiencies, the one that I was alluding to in the previous ones. There are transporters, because transporters are required to circulate or move these nitrogen-rich compounds in the generation of urea. And then there are secondary defects in the urea cycle, which also can lead to hyperammonemia. Generally, urea cycle disorders are quite rare disorders, most common being OTC deficiency, somewhere between 1 and 60,000 prevalence, to some of the deficiencies such as NAGS and HHH, which have a very low prevalence, meaning they are ultra rare. The amazing fact is, within a decade of the diagnosis or discovery of urea cycle disorders, the pioneering work of Saul Brusilo, Mark Batshaw, Dave Valley, and many others started to formulate therapies. Again, the two ways they tried to address this is because ammonia comes from waste nitrogen. What if you give dietary therapy to decrease the amount of nitrogen load by restricting with protein? So that was the dietary therapy. And what if we devise an alternative route? So if your main highway is blocked, you have a local road that you can take to. Can we use a local road to divert the traffic away from the freeway, thus reducing the congestion? So those were the medications that were discovered. So before the discovery of the medications, the outcome for individuals with urea cycle disorders was rather bleak. The 1 year survival was 14%, and vast majority of survivors had severe IDD. This is from a classic paper from Vivian Shih from the 1970s. But even soon after the availability of nitrogen scavenging agents, the outcome moved towards the positive side. In this publication by Masal, Batshaw, and colleagues, they studied 26 children with UCDs, and the 1 year survival improved significantly. However, a vast majority of them still had intellectual and developmental disability, and the average IQ in that study was still in the very low ranges. So the burning question over the next few years became, how do we improve the outcomes? How do we transform the care for these individuals and families with UCDs? Now, a challenge in doing something like that is that UCDs are rare disorders. So generally for all rare disorders, including UCDs, the diseases are not well characterized. We do not know what their natural history is, what are the organ systems involved, how to pick out the ones that are going to be severe from those who are not, and so on and so forth. Rarity also means that there are few expert centers for management and research. Again, many of you have lived experience with this, and I don't have to tell you all that. Aha. My phone just banged on the slide, and maybe that's why you lost what I was sharing. Are you all able to see the slides again? Okay, perfect. So recruitment for trials is usually difficult, and the study populations are quite dispersed. So this often leads to treatment without high quality of evidence. So how can the outcomes for people with UCDs be improved? That will take a amalgamated effort of research, improved laboratory research to identify targets for treatment, clinical research to develop these therapies, collaboration with industries to get these medications to approval and make them available to all of you. It needs public policies, which the NUCDF has championed, and I'm going to talk about that later. How do you diagnose these by newborn screening? How do you provide incentive for industries to develop these drugs through the Orphan Drug Act? How do we make genetic testing available, and how do we prevent discrimination against use of that? The GINA Act of 2008. How do we improve clinical care by diagnosing, appropriate therapy, and access to care? And finally, how do we disseminate all of this knowledge by educating everyone who's required to improve care, the families, the providers, as well as the next generation of investigators? So all of these would be required to improve. And the one key glue that holds all of these facets together are y'all. All of the patients, all of the families, and the patient advocacy groups who move every important facet of this wheel forward. So what is your role in transforming life for people with UCDs? And how does your participation, though it might not be of direct benefit to you, impact the overall field and the community and make an impact on people you have never met or will never meet? So by providing direction for research, you are guiding the entire research community on what should we focus. By involving in research participation and research advocacy, you're moving not only academic trials ahead, but also industry trials ahead that get better therapies. By being involved and being the voice of the people who vote, you're championing the laws that will bring public health policy changes to improve healthcare within our country. You talk to physicians by telling us what works and what does not. And I hope most of your physicians listen to it, because the people who are leading the field certainly do. Our direction of research and where we need to go is very much informed by your voice. And finally, by allowing all of us to be educated on what is important, you're doing an important service by training the next generation of scientists and physicians, and you'll see one of them who will give a talk today later. So patient advocacy and patients are at the center of what has been transforming the care in UCDs. There are multiple patient advocacy groups that I have listed here. I can't list all of them, but there are many of those, and I've listed the ones with whom I've had the privilege of working, most importantly with NUCDF. So because I'm a lot more aware of NUCDF's role, I just want to take you through the story of how they have impacted the field. In the late 1980s, NUCDF was founded only by an initiative of a few families that came together and said, we got to do something to move this forward. Within a few years of its inception, NUCDF championed for the development of sodium phenylbutyrate. Those were different times when the medication was being compounded at the pharmacy of Johns Hopkins, and the funding was about to run out. And it is possible that and it's very likely that without the activities of NUCDF, that may not have gotten across the finish line to be the first drug to be approved. In the early 2000s, when the Rare Disease Advocacy and the Rare Diseases Act led to the establishment of the Rare Diseases Clinical Research Network, NUCDF was right there. It became a pivotal member to establish the Urea Cycle Disorders Consortium, which at that point had 5 to 6 sites, and the UCDC assisted in the startup, and with the O'Malley Foundation of philanthropy, we were able to kickstart this consortium. When the consortium kickstarted and we were advancing some of these studies, the NUCDF was pivotal for the growth of the sites. We went from 16 sites to currently now 17 sites. The NUCDF established a fellowship that allowed multiple individuals, including myself, get protected time to continue our work and remain within the field of the UCDs. Finally, now, after 2 decades of consortium work, when the NIH funding is ending, NUCDF has taken on a broader role to see how do we democratize all the advancements we have made, how do we take it further, how do we develop comparative efficacy research and networks so that people get care across the wide geographic expanse of our country. And you're going to hear a bit more about that in today's session. So thanks to the philanthropic funding, the researchers, the staff, the patients, the NUCDF, and others, the UCDC now has 17 sites. The most recent one which I don't know why is not listed here, is St. Jude at Memphis. Now, in spite of us having a big expanse, you do see that there are many gaps within the Midwest, the Southeast, and sort of the Northwest regions of our country. And again, I'm not an expert to talk about the expanse in the other parts, but I would say that there are large holes in expertise care across the continents. So what has these collaborative networks that have grown because of patient advocacy done? At least the UCDC, I can talk, has implemented 19 studies that have collectively enrolled nearly 1,700 individuals. And these have conducted longitudinal studies, randomized clinical trials of novel medications, biomarker discoveries, comparative efficacies. The PCORI liver transplant versus conservative management was indeed led by the NUCDF and so on. There's been a lot of studies that the UCDC has been able to do. Just similar to the UCDC, a lot of other consortia and family foundations and networks have developed. So these include the European Registry, a network for intoxicating metabolic diseases, or EIMD, that has enrolled 700 individuals with 40 partner sites in Europe. There's been the Citrin Foundation out of Japan that is looking at citrin deficiency, which is relatively common in individuals of East Asian ancestry. And there are liver transplantation consortia, all of which have made multi-center, collaborative, large-scale efforts possible. And without this, we would not have moved the field forward. All of these consortia have led to a significant increase in research in the UCDs over the past 3 decades. If you were to look in the 1970s and 80s, there were relatively fewer publications that were geared towards the UCDs. And as you see, past the 1990s, there's been a significant increase and interest in UCDs. This trend would generally be true for most things. I hope as humans we're growing to be more inquisitive, smarter, and better than what we were 30 years. But this inflection point is really impressive for a rare disease such as UCDs. More importantly, if you were to look at how many clinical studies were listed on clinicaltrials.gov between 1990s and 2006, the only things that I could retrieve were a handful of studies. Now you have 77 studies that were listed as of last year. And out of those, a majority of them were intervention, where they were looking at a methodology to improve the care of individuals with UCDs. And as Tresa alluded to before, now in the realm of genomic era, where we're having outstanding diagnosis and genomic therapies, newer industry-led genomic therapies are also being investigated. So with all of this, where are we now? We now sort of are able to diagnose earlier. We have potentially better biomarkers. We have better treatment protocols. How has that been transformative for care? So if you look at some of the data, it's complicated the way you look at data, so I will not make dogmatic conclusions of it. But when we have looked at the data from 503 individuals with UCDs from North America and Europe for the distal UCDs, ASS1D and AS, the citrullinemia and argininosuccinic aciduria, you see that individuals diagnosed with newborn screening tend to have higher cognitive scores as compared to those after symptoms. While current standard of care, when you tease it out, we are still not, with the sample size, able to see significant differences within the neurocognitive outcomes in the subset of OTC deficiencies within this database. So where are we now in the collective efforts to transform the lives of people with UCDs? We've had significant increase in federal, philanthropic, and industry funding that has allowed for investigation and or approval of new treatments for these disorders on the research realm. Newborn screening has become the norm in 50 states of the United States. Genetic testing is being covered by more and more insurances, and many times by state-funded insurances across. There's continued and growing industry interest given the public policies that have been in place for drug development. From clinical care, the early newborn screening as well as genetic testing is allowing earlier diagnosis. The better education and outreach is allowing for clinical diagnosis of individuals who are missed. And the therapies that we are using and the protocols that we are using are making them more effective as compared to what we were before. And the amazing outreach activities of NUCDF and many consortia are moving family provider education to the next level. So if we are all so good, then why do we need to improve? Because we are not all so good. We need to democratize care through wide outreach. There are large gaps in how the care is delivered, and even at centers and places of that have enough resources, there is no equitable care across. So how do we do this? How do we educate people? It is by developing resources, by developing educational materials that can be asynchronously tapped into, especially with the current generation who like to view rather than read many of the resources. The UCD teleecho, again being led by the NUCDF, will be an important next step. So you're going to hear about these in one of the talks that the NUCDF is going to give. We need to continue our investment in the young investigators, especially in the challenging research funding scenario. And you'll hear from one of our younger investigators as to what that investment looks like. From the clinical standpoint, given that we now have widespread genomic testing, we're uncovering variants of which sometimes we don't know what to do with them. So we need functional studies to understand this genetic variants for earlier diagnosis. Dr. Amy Dudley is going to present her wonderful yeast model as a talk within the next hour. And Stephen Lang, who is a trainee from Baylor, will tell you how he collaborated with Dr. Dudley to look at the functional impact of two common variants. We need improved access to care and improving education. Dr. Andrea Grokman is going to give her talk about how you should recognize UCDs in individuals within the ER who present with neurological symptoms. And she's going to give an elevator pitch also for where we're going to go from here for the clinical care networks. Now more than ever, we need public policies that further research, advocate for individuals with IDDs, and foster affordable treatments and make them available for all. You will hear some of this from the NUCDF's talk on their next roadmap in the next 10 years. And finally, from the research standpoint, you need better monitoring of diet and ammonia, so the Rare Diet app and ammonia meter, home ammonia meters, monitoring for complications such as liver disease, novel therapies, including genomic therapies, gene editing therapies, protein therapies, and small molecules, which are new emerging therapies that Laura Conzel is going to talk about. And when we have to do such, how do we have accessible research networks so that these could be implemented easily across the United States? That, again, will be the focus of our talk. So you're going to hear all things that we have to do in the talks today. And finally, how do we think we're going to get to where we want to go? This, again, is a roadmap that has been put forth by the NUCDF Partner Network, where all stakeholders will come together to participate in future comparative efficacy research, comparative equitable care delivery, and education. So I want to end my talk by thanking the wonderful UCDC team in which I grew up with and whose work I presented in this talk. This is the current and previous leadership, the NIH partners, the program staff, the patients and the patient advocacy group, and most importantly, the research staff whom with you interact. They're the engine who drives everything forward for us. I would be remiss if I did not acknowledge these visionaries, including Cindy Lamons from the NUCDF, who started all of the UCDC many years ago and then had a roadmap so that the current leadership could take over and move further the good work that they had initiated. And I want to thank the UCD A team at Baylor College of Medicine that has taught me everything that I know about UCDs and continues to educate me about that. And at the end, I want to thank Cindy Lamont, who's one of the prime reasons as to why I got into the activities of the UCDC and still remain there. And I don't know whether Tresa is going to like this, but I want to thank for her taking over the leadership and doing 4 jobs at the same time and leading the vision of UCDC. And most importantly, I love showing Lexi's picture. Tresa said her first job is to be Lexi's mom. My first job is just to do what Lexi tells me, and the remaining time I have, I do the rest of the things I said. I really want to thank you all for your time, and I'm happy to take any questions.